Hello, thanks for joining this session. I'm Kadryn Kadasia. I work for Particles for Humanity, a public benefit corporation operating out of the greater Boston area. I'd like to start by recognizing the conference organizing committee for this opportunity to share some of our findings on a technology we're developing to improve rabies vaccine access and compliance in low and middle income countries. I know this audience needs no introduction to the harmful effects of rabies on human life, despite it being a fully preventable disease. The two current approaches to rabies vaccination, that is pre-exposure and post-exposure prophylaxis, are encumbered by prohibitive costs and loss to follow-up, which limit widespread uptake in certain settings. As such, single injection vaccine delivery is predicted to have a significant net positive effect on rabies disease management. The technology we're developing comprises degradable polymer capsules delivered in a single injection that mimics a multi-dose vaccine schedule. Vaccine is loaded into the core of a particle about the size of a grain of salt, as is shown in this graphic on the top right corner of the slide. These particles are made from a water-degradable polymer using a unique process that results in a sharp burst release of the vaccine. The idea is to then take different polymer compositions with varying degradation time points and combine them into one injection that mimics a multi-dose vaccine schedule. Here's a schematic that better explains this concept. Starting off with three different polymers indicated here in the red, green, and blue, each of one of them degrading at a different time point, say for example, day 7, day 14, and day 21. We can manufacture the particles shown above from each of these polymers, load them with vaccine, then deliver all three particles in a single injection. The vaccine is predicted to release on day 7, then again on day 14, and a third dose will be released on day 21, resulting in this cumulative dosing profile equal to if the individual had visited the clinic on 3 separate occasions for 3 different injections. There are some challenges associated with this technology as is. One relates to injectability based on the particle sizes, vaccine stability, process scalability, as well as cost, all of which we plan to take into consideration during further development. Here's the process by which Particles for Humanity currently manufactures these particles. On the left, we have a cartoon depicting the process, and on the right, we have a real-life image of either the outcome or the equipment in use. We begin with polymer pellets, which are thermally cast into a film. The film looks exactly as is shown in this image. That film is then micromolded into bases that have an open cavity on one face. And so here we see the particle bases with the open cavity. The cavities are then filled with a liquid payload using a high throughput liquid dispenser, and the material is left to dry under ambient conditions. So here you can see the tip of that liquid dispenser, and you can make out the particle bases on this slide. We can either fill the particle bases with a model fluorescent compound and plan to develop this into formulations containing the rabies antigen. The filled bases are aligned with caps made from the same polymer types, and the particles are sealed by heating the contact surfaces, and that's shown in this image here on the right. Finally, the particles are manually isolated from the film and singulated, resulting in these single particles, as we can see in the image. The overall process summarized here allows us to generate material for subsequent in vitro and in vivo studies. One such study is to look at in vitro release of payload to confirm that the particles exhibit the predicted degradation profile. Shown on the left side of the slide are particles filled with a fluorescent molecule, and on the right are the data generated in an in vitro experiment where we detected fluorescent signal on day 6 from these particles, indicating release of the payload. Building up on these preliminary observations, we've partnered with global manufacturers to source rabies vaccine for continued product development. We've also obtained commercial materials such as the reference vaccine and the 7th International Standard for Rabies Vaccine which we use to validate our protocols. Meanwhile, we established relationships with contract research organizations through whom we conduct our antigen characterization studies that I will highlight in the upcoming slides. We used transmission electron microscopy, that is TEM, to visualize purity and morphology of the virions in these rabies antigen samples. As shown here, the reference product yielded the classic bullet-shaped appearance of rabies virus. The first antigen evaluated was not viable, with only fragments observed under TEM. The second and third antigens evaluated appeared to contain numerous intact virions of the expected size. And these microscopy data altogether enable early selection and prioritization of antigens for further development. Next, we used nanoparticle tracking analysis, or NTA, to measure the size, homogeneity, and distribution of virions within these antigenic samples, with an expected primary peak around 150 nanometers for intact antigen. This was observed in the commercial reference shown on the left, as well as the second antigen evaluated shown on the right. However, in the first antigen evaluated, we saw these multimodal aggregates confirming some of the observations that we saw in TEM. We also have plans underway to complete NTA for the third antigen that we have received. Next, we established an in-house ELISA protocol to assess the baseline stability, the baseline potency of the antigens that we received, using the 7th International Standard for Rabies Vaccine to generate standard curves for quantification. The data that we obtained are shown in the graph here on the right, where we detected a potency of about 30 IU per ml for the commercial reference. The first antigen evaluated came in at about 10 IU per ml, which is significantly lower than the reported value from the manufacturer. The second and the third antigen evaluated both had a reported IU similar to what we detected, and so these are confirmed choices for further development. The planned future work on this program will be to address potential shortcomings of the technology, knowing that we need to look at further analysis of the rabies antigens to mitigate potential loss in immunogenicity associated with this single injection technology. So we've planned a two-step approach to this, beginning with pre-formulation development, looking at temperature as well as pH-associated degradation, and mimicking some of the processes that the particles are subjected to, such as sealing at a certain temperature, as well as storage of the particles, both in our experimental process and in vivo. And we also know that there is a local drop in pH within the particles during degradation, and it would be important to assess the stability of the antigen under all these conditions. We'll then build that up into stability and release studies, looking at various particle compositions and levels of ingredient loading into those particles, improving the process efficiency, looking at storage, lyophilization, and freeze thaws, and then continue with our analytical characterization and formulation development. One thing to keep in mind is the broad applicability of the single injection platform, with potential for large impact in many therapeutic areas that require multiple dosing regimens. And as a company, we're keeping all these possibilities on our horizon for long-term development goals. I'm going to conclude by thanking all the individuals who have supported this work, starting with the PFH team shown here in the top half of the slide, our extensive network of consultants, our research partners, and the Gates Foundation for providing the funding for this program. Thank you, and I'll be happy to take any questions.